Can you guys, everyone can hear me okay as well? I'm going to talk in this volume today. So, all right, let me share my screen and let's get started. Is my screen being shared? I have no idea. Okay, hope it is. Okay, so let's get started. We'll just go through this pretty quickly. So thank you all for joining this year's BioClub Lab Safety course. Um, Unscripted, you can probably tell, but let's jump right in. So today, just want to cover some of the basic types of lab hazards that you might encounter during your experiments. You have chemical or biological hazards, electrical hazards, temperature hazards, both hot and cold, but for BioClub, maybe just hot, and cutting hazards. So this is not an exhaustive list of all the types of hazards you might encounter, but it's just kind of the most common and I think the most relevant to our lab. So I will be tackling these mainly. Oh, whoops, there's something on here. Hold on. Is this picture showing? I hope this is. Okay, I hope you all see this picture. I wasn't showing up on my screen for a bit, but so this is a picture of our lab, and as kind of things goes, we have, as you know, glassware on the top left hand corner of the pictures when you enter into the lab. You also want to be aware of any ground clutter when you walk in and proceed further into the room. We also have a number of electrical, these, what are they called? Power outlets or power taps. I don't know what that is in English, but sort of these things. So you want to make sure that they're in their properly seated. And of course, we have the autoclave, which is right next to the door. Um, so one thing that I do when I walk into the lab is these are kind of key areas within the lab that I think everyone should be kind of weary of or just kind of, be suspicious of, if that makes sense. Um, so that means like, assuming that the autoclave is really hot, even though it might not have a green lamp on, might be really just be aware of that. So don't put your hand on the metal bits. Um, Glassware, might be, on the way to falling or just, have fallen and there's, shards on the ground, for example. And these electrical outlets might be, might not be, they might be really dusty and they could be a potential fire hazard. So what I like to do is just kind of keep a mental note each time I go into the lab and of course when I walk out the lab. So that's something I encourage you all to do. So today I think I'm gonna just briefly divide the talk into two sections. First part consisting of preventative measures, and then in section two, I'll go over what to do in case something happens and we'll have a primarily focus on the preventative measures as a lot of these things are preventable. And I think um, this is kind of the most key part of lab safety. So section one, preventative measures. One thing I really want to kind of drive home today is that the first line of defense is proper handling. Now, when I mention handling, I don't mean just things you can hold in your hand. I'm also talking about things in your environment as well. And so why is, handling the first line of defense? Well, there's an argument that one could argue that even the highest tiers of personal protective equipment or PPE, which I'll talk about in depth a little bit more, is not foolproof or error proof to humans as well. You can have, as picture on the right shows, you can have the mask, the eyewear, the gloves on, but if you touch something, with your hands, you're not supposed to, and then go scratch your nose or touch your face, then go under the mask and touch your face, that is, PPE won't protect you from that kind of human error. And also these PPE, they're meant to solve specific problems or hazards, prevent specific problems from occurring. Gloves, for example, might be good at, getting your hands, it might help you from preventing your hands from getting dirty, but not against heat, for example. And so for these reasons, I'd argue that being aware and really understanding what you are handling what is in the lab is should be the first line of defense. Uh, Do's and don'ts of basic lab safety. Do again familiarize yourself with the reagents that you're handling. Definitely consult the operating manual for the machines, analyzers, and things like that before you even touch them. And if you're unsure, do definitely ask experienced lab users like me or someone else who are in the area. You can ask Discord as well if you are unsure, and just double check that you're doing the right thing. Um, the don'ts, we go on the right hand side here. The don'ts: do not swallow things that you've touched in the lab. Don't touch something and then again, touch exposed areas of your skin and face. Don't leave, samples and reagents unlabeled and just leave them in the lab. And also, really important, don't conduct experiments at the lab when you're feeling unwell. Uh, a lot of these, seemingly obvious things can happen when you are, you know, under pressure from a deadline. You're stressed out. You've only got 2 hours of sleep. Um, or you're running 10 experiments at once. Please don't do these things. Just don't do it. A lot of these preventative measures are about de-risking. And so in line with that, make sure that you are coming to the lab with the best kind of conditions as well. Other things before you start your experiments, please wear PPE as intended. I'll cover this a little bit in the later part. Make sure that your work area is clear, and be mindful of the other hazards that we kind of talked about: the electrical outlets, open flames, combustible materials, and other sharp objects that may or may not be, hiding in plain sight, so to speak. Especially with electrical outlets, you don't want to daisy chain these power taps together, and just make sure that there's no dust accumulation and things like that. As for, we have a picture of a Bunsen burner here, but actually in the lab, we do have what do you call it, like a torch, like a gas torch, methane torch, I think, or some sort of gas torch as well. So that's not the only kind of potentially combustible thing that we have in the lab. So please consider these things as well. Here's a painting here of an alchemist. These are just an example of what you should not be doing. I mean, you have this, puppy on the bottom right hand side of the corner. You have open bottles and, ground clutter around the workspace. You have an open flame here and questionable degree of ventilation in this environment. And you also have, these people in the back trying to, get this person's attention. And this is just not a great lab environment. These are all kind of potentially hazardous. Here's another picture here. This person is, again, eating in the lab. Please don't do this as well. They have these kind of glassware filled with, this is obviously just a demonstrative picture, illustrative picture on purpose. And, there's stuff that's not cleaned up, spills that are not cleaned up. There's things like about to fall over, open-toed shoes, or sorry, no shoes. And yeah, this sort of overflowing liquids. Yeah, in general, you don't want to fill it up to the brim, even though it says 500, milk capacity. Don't try to push that to 600, or I just don't recommend any of this stuff as well. Okay, so briefly covering the sort of general things about general things you can do to prevent these common types of lab hazards. Just to go over with the chemical and biological stuff, which I'll go into depth in a bit. Please do understand what you are working with. As for electrical hazards in the lab, check for the loose connection. Don't daisy chain your power taps. Your temperature hazards, just assume that the autoclave is hot. Don't lean on it and just assume that it's not okay to touch with your hands. And of course, cutting hazards, especially with glassware we have in the lab, they might be broken on the ground. So please check for those as well. And some of us might be working with microscopes as well. So the cover slips and those things are, of course, will be a part of potential hazards for related to cutting hazards. Okay, so now I'd like to kind of shift focus. So these are kind of in general things I'm sure you were aware of at the house as well. But now I'd like to kind of go deeper into the chemical and biological hazards. So the next section will be kind of considerations towards chemical and biological hazards. Um, Cover our bases here. Well, first things first, washing your hands when you enter the lab and when you get out of the lab. Uh, you know, Ideally you would have warm water. We don't have warm water in the lab, but that's totally fine. Actually, the CDC doesn't it doesn't say it has to be warm water, but please do wash your hands with soap, um, maybe like 15 seconds of vigorous like hand washing, um, and do this before and after leaving the lab. Glassware utensils are for laboratory purposes only, lab use only. Don't, not that you would ever like maybe do this, but don't, prepare, consume food and beverages using the glassware um, in the lab for human consumption at least. And of course, keep your workspace clean. If you spill something, clean it up immediately, dispose it trash into respective bins. And this is kind of outside of today's the scope of today's lecture, but just if you touch anything that touches genetically modified organism, it must be autoclaved. So if a pet tip or some plastic or something or glassware touches this modified organism, you gotta autoclave that. And then only then can you take the trash outside the lab. So be aware of those things. Next, we'll talk about exposure routes. Again, not an exhaustive list, but common ones: absorption, inhalation, ingestion, and injection. Goes without saying, please don't inject yourself here at the lab. Not that we have anything that you can inject. And just know that these are some of the routes that potential things can happen with. So, especially, going back to the thing of not touching your face area, inhalation and ingestion. These are the most common routes as well. And then now going to PPE, personal protective equipment. Please do consider this as your last line of defense. Don't rely solely on PPE as for the reasons mentioned earlier. They're not foolproof. They're not going to protect you from all the things that could go wrong. But PPE is exceptionally good for dealing with chemical and biological hazards. So obviously these things kind of help you from prevent you from getting dirty and unwanted things on your clothes and on your person. But they're also really great at protecting you from the invisible hazards, including microscopic organisms and aerosols. With masks, there are different types of masks out there. The ones you buy at store are perfectly fine. If you want, you can get N95. That's a bit overkill. You can also double up masks as well. So if you're feeling, in particular, you can definitely do that. Eyewear, if you have glasses, that's totally fine. Actually, at my work, I always show up in glasses, so I'm not, constantly worried about getting something in my eye. But if you do want protective eyewear, maybe we can procure those for those who want it. I think I have some in my home as well, so you can borrow that. Just let me know. Gloves, wear them, as they were intended. You have nylon gloves. We have nylon gloves in the lab, and there's also latex gloves. Nylon gloves are, in my opinion, the superior choice. They are resistant to um, just an all-around very resistant to different types of chemicals. Uh, okay, we have goggles in the lab. Okay, thank you. And lab coats. I think we have some lab coats, but yeah. Also, if you come in socks or or wear slippers, the slippers in the lab, please. What else? I know it could be difficult, but also try to wear long pants as well. But lab coats, yeah, for sure. If I mean, I've gotten stuff on um, my clothes that I can't ever fix now. So if you, yeah, maybe something to consider as well. Don't come with the most, your most precious items of clothing. A little bit more things about chemical hazards I want to talk about. So, um, w- so I use the word reagents here. Reagents kind of refers to all the, things in the cabinet we have, whether that be for preparing media for bacteria or the kind of like ethanol and methanol that we have in the lab, I don't know if we have methanol actually. Um, but also there's just, that cabinet of just bottled reagents, powdered liquid, but especially for um, liquid reagents, you want to prevent the formation of aerosols. So what are aerosols? Aerosols are loosely defined as very fine solid or liquid particles dispersed in gas. So in this right side, the picture of the right side, this is coming out of a canister. This is an aerosol. Uh, and so you can accidentally create these by the kind of mechanically as well. If you're pipetting the wrong way, for example, you might have some very, very like kind of some form of like this is again this is like a form of spilling, if you will, but it instead of going to the ground, it goes up in the air. This is particularly dangerous if you're working with highly acidic like sulfuric acid comp like reagents like that. You can maybe when you're pipetting or something, you might accidentally creating a lot of bubbles, and then you're accidentally creating few like aerosols that go into the air. Again, same principle with gas as well. If you're warming something up and um like this here, and you got all this steam generating, if you're uh I don't know, you would never do this, but like if you warm up gasoline or something on a hot stove, and then the room is filled with combustible, some like that's a little extreme, but those are some things that can happen. So um, please be really sure that the thing that you are working with is okay to warm up, and you can find information on specific things online, and and please do consult me, and we can figure something out together. We find ways together. Also, another aspect of chemicals is that some compounds are volatile, so meaning that if you leave open a if you leave the container open, it'll just start escaping the bottle and just start accumulating in the air. Again, creating a similar effect of having generating gas into the room as well. So, you have a room filled with, gas gasoline, and you light a match, then that's very dangerous. And also, depending on what you're it might not be, explosive in nature. You might be filling the room with nitrogen, or you can't smell that stuff. So anyways, for those of you who didn't know, please do be very careful with these things. um One, I think, more common thing would be like, you're moving liquids from one container to another container, and you're going from like you're dropping the liquid from super high up, and you're creating all this like you're splashing liquids around. Please be mindful and be careful when you're handling these sort of liquids. Um, so as for kind of the aspects about understanding what you're working with, again, don't use unlabeled materials. Please label the things that you're working with. Sometimes you have a original, the bottle is something that a good practice might be, you have a bottle of reagent, a liquid reagent, and you want to, you only need a little bit, but you want to aliquot or put it into a smaller tube just so it's easier to handle. And then, if you don't label that, then, no one knows what it is anymore. So please label all the things that you've aliquoted. Again, if even if you think it's safe, you should assume that it's the worst, and assume the worst and practice proper handling. Again, don't leave containers open. You can prevent spills and cross contamination, and prevent those volatile compounds from going up into the air. Um, I'm giving you this kind of worst case scenario type thing because I mean, as long as you treat them as is, as if they are kind of really, much more worse than they are, then you will be fine. You will be more confident in dealing with these sort of more, how should I say, like dangerous chemicals. Not that, if not that I think that we will use them or that I think you will use them at some point, but just in general. So it's how to look up these sort of safety related information and very specific things. Please consult the safety data sheet, SDS. This is required by all companies that make these reagents they have to make these by law. So it'll have information on how to store these things, how to handle them, how to dispose of them, how to provide medical treatment, and other potential hazards. So, and it's as easy as, for example, methanol, SDS. Just Google that. You will find you'll find PDF links, and different companies will be providing that. It just, if the company A is making methanol and also company B is making methanol, that's the only difference between these things. They are standardized and regulated. So please do, uh, just for your own, knowledge as well. You can learn a lot from these things. So please do try this if you have time. And if you don't, we'll do it together. Next things, biological hazards. Just a kind of sideline here, but also sort of related, I guess. The distinction between sterilization and disinfection. Sterilization is by definition a sterile. So I'll read this out here. A sterile surface object is completely free of living microbes and viruses. Sterilization procedures kill all microorganisms, and the method that we use is mainly the autoclave. Disinfection, on the other hand, by definition, as stated here, eliminates most pathogens, but not necessarily all types of microbes. Disinfection reduces the level of microbial contamination. Chemical disinfection does not kill spores, unlike chemical sterilization. So there's a difference. I guess they're going into the difference between chemical disinfection and chemical sterilization. But either way, for example, when you wash your hands, that's a type of disinfection. When you're dunking your glassware, for example, in 10% bleach, or wiping down the workspace with 70% ethanol, just know that it's not going to completely remove all the microbes. Like I said here, there's some spores. There's a spore forming bacteria is a kind of common thing that microbes do, I guess. And sometimes they do cause disease like Clostridium, but yeah, just so we know that there's a difference between when we talk about sterilization and disinfection, just so you know that there is a difference here. Just a little sideline here on disinfectants. So for example, why do we use 70% and not 100% ethanol? So ethanol this is the chemical structure on the top right. It has this OH sort of oxygen and hydrogen here. This is what makes it an alcohol. So anything with an OH sorry, it has to be bound to this carbon and it doesn't have anything else. It just has hydrogen. But if you see an OH, you can, by definition, the chemical definition is that it's a type of alcohol. So ethanol, with this sort of disinfectant works by neutralizing proteins. And what it does is that you have like a protein and then the ethanol comes in and it breaks the chemical bond that maintains that protein structure. For those of you who know, probably know the protein function of, like, let's say it's an enzyme, is encoded in the protein structure. So basically it breaks down the proteins that living things need as well. But the problem is that pure ethanol can't penetrate the bacterial cell wall on its own. And what the water does is that it helps that ethanol kind of permeate through the cell wall and then destroys the bacteria from within. It destroys the proteins that the bacteria carry from the inside. And, in this case, we use ethanol, but same with bleach, for example. You use 10% bleach and not 100% bleach. So these are some just kind of general things to keep in mind when we talk about these sort of disinfectants. Yeah. Um, a little bit about sterilization. So again, sterilization is the one that it kills all microbes. It's 100%. Everything is completely dead. Uh, just a reminder on how the autoclave works. Note that it's a function of heat, time, and pressure. Of heat of around 120 degrees with time to 15 to 20 minutes and a pressure about. I actually don't know. Actually, I have to look that up. Sorry, but with higher pressure than normal atmospheric conditions here. So what it is that it combines kind of this different. So you can kill things with microbes with just heat. Maybe not just time, although maybe they'll eventually die, but they'll proliferate. So never mind. You can also, I guess, kill things with high pressure as well. But basically, the idea is when you combine different methods, you will result the results in higher confidence levels of sterilization. And so this is kind of the main difference. Like just another concept that could be applied to disinfection. So in disinfection, we also have different levels, like low and high levels of disinfection. One thing you can do is consider using hot water over cold water and soak objects in disinfectant or soap longer. These things all contribute to a more thorough disinfection process or cleaning process. So this is just a little sideline, but I thought it would be important to share just so you guys know. Okay, so kind of wrapping up here. Just want to talk about what to do in case of an emergency or something does happen. Dealing with cuts, I know we're all adults here, so it should be okay. Please rinse off. Step one, please rinse off with water, stop the bleeding, then apply disinfectant, and this can be again ethanol or hydrogen peroxide. Apply bandages, and then please report to lab manager Gina, and if needed, seek further medical attention. Again, I talked about a little bit about exposure related hazards, but now if something were to happen, most cases, just rinsing off with tap water is going to be the way to go here. Let's say you are handling and you didn't have gloves on for some reason, and you spilled a little bit of hydrochloric acid or something. We don't have this in the lab, but just for an example. So if just touching it a little bit isn't gonna like immediately cause pain. I mean, if you get it on your fingertips, it might sort of what do you call it? Like eliminate, like get rid of your fingerprints. I kind of like that, but basically it's happened to me before. It's not like super painful or anything. It doesn't feel like anything. And what you're gonna do is you're not gonna try to, if you have acid on, you're not gonna try to neutralize it with a sort of base. You're just gonna dump water on it as much as possible. So you just go to the sink, open the tap water and just rinse it off. That's probably the most like just the default thing that you're gonna be doing. If you accidentally ingest something, this one can get a little tricky because it depends on what you ingested and how you did it. But depending on what you ingested, don't induce vomiting. This is because your body produces stomach acid. It's acidic, and if you try to reverse it back up, you're gonna end up damaging the soft tissue in your throat, for example. So just go straight to seek medical attention. Grab somebody, explain someone, and then or just call Gina or myself, and we will figure out a way to get you the appropriate medical attention. Inhalation, again, this is sort of vacate the area immediately, and then seek medical attention, similar to what you do with ingestion. Basically, this, again, this is most unlikely to happen, but in the unlikely situation that something does happen, just, be aware of what be aware of sort of just what you should be kind of doing. In case of a fire, again, inform people in the area to evacuate. If the fire is small, waste basket size, please acquire a fire extinguisher and extinguish the fire using this. Oh wow, I've written fire extinguisher 3 times. Anyways, it's located on the second floor. The next time we all come in, we'll go over where it is. Warn others, and if it's really out of control, dial 119. This is not the police. This is the fire department or emergency services in Japan. So 119. Please remember that number. And of course, evacuate and inform the lab manager as well. I think that kind of briefly covers, basically covers all the things that you might be dealing with. But just a reminder here, before you start your experiments, check your surroundings, check those key areas that I highlighted in that picture. Make sure your workspace is clear of clutter. Don't, have stuff out that you need in the like kind of immediate future. Don't have other stuff. Try to like not put stuff that you don't need in that workspace. And then of course, check in with your lab mates. Let people know that you are in the lab and what you're doing. And yeah, communication is a key here. Working alone, please understand the risk. It means that you are either out of direct visual contact or hearing range with another lab worker for more than a few minutes. If you have to work alone, please have you considered the risk, to your physical health? And do you really understand the materials that you are working with? And if it's your first time, you might not have a lot of experience. So if you are unsure about these things, please refrain from working alone. If you decide to work alone, anyways, either way, you're gonna please let other people know in the Discord. Wrapping up here, some things with housekeeping. So no shoes in the lab, no food or drink in the lab. Please, don't let certified people just alone in the lab. Glassware, liquids, and heavy items should be stored on lower shelves. Immediately notify lab manager on Discord for broken, malfunction equipment on glassware or shared reagents that are running low. Probably sort and dispose of trash. When you leave the lab, just remember to ask yourself these questions. You cleaned, wiped down your work surface? Have you washed the glassware and put it on the drying rack? Have you replaced any equipment that you use? Do you put your name on the incubator log? I think we haven't developed this yet, so we will in the future. Take out the trash. Did you properly turn and run the autoclave successfully? Please turn off the lights and any other unused equipments. Log your entry time and exit time on Discord, and please do lock the door. So lab access. This is the link to the lab safety quiz here. Maybe we can send this in the chat as well. But so get a perfect score. You can take as many times as you need. Notify Gina that you've passed in this lab safety thread. And as you know, digital keys are managed to the Curio app. And once you've been granted a key, you will receive a confirmation email with a link to download the app and instructions on how to set up your account. So please do this properly. I know I got this a while back, and then like I don't know where that email went, and I like didn't know what to do. So that could be a little, yeah, just ask for it once, and then it should be good. This is the end of today's lab safety lecture. I hope you guys, I hope the presentation was somewhat understandable, but last thing, lab safety is, just from my experience, lab safety is a cultural thing as well, right? You can't, it's not a specific to-do list, but it is also has to do with um, with the kind of labs culture. If we set a standard of lax and we're bending the rules all the time, then everyone else is gonna be like that, unfortunately. So especially with today's members, I see you guys as like the future, and on still leader into the leadership within BioClub. Um, please don't cut corners. Uh, you know, when you slack off, everyone else sees that, and people are gonna like, just gonna be bending the rules all the time as well. So please set an example and set that standard really high. I have I do have high expectations for you all here today. So yeah, with that in mind, I think we will end this portion of the lab safety lecture. Gina, was there anything that I missed, or do you want to take over, or things you want to add?
Oh, well, first of all, thank you for delivering that great presentation. I think it was very clear and concise and very easy to understand. So thank you. Yeah, I think the only thing I would add is, of course, the Curio, the digital key process, both for the people here and people possibly watching the archive video in the future. I'm very accessible on Discord. So if there are any further questions about downloading the app or setting that up, I'm more than happy to answer those questions and figure that process out. In addition, I should be getting emails whenever people submit the quiz. I should be getting them, but again, please contact me in that lab safety thread in order to confirm that I have received that and that I know that you get a score. That way I can get you that confirmation email as soon as possible. Yeah, but I think that's about it. Yeah, just again, it is a cultural thing, and I know that many of us have been using the lab for a while, referring also to people who watch this from now on. And I think that we have kept it pretty safe so far. As far as I'm aware, there have been no big fires and nobody's had a serious injury in our lab, which is great. And we really want to keep it that way. And yeah, it's all about a community lab, right? So being able to keep ourselves safe, keep each other safe, keep each other accountable, so that we can all continue using the lab and doing some cool stuff. So. Thank you very much for attending.
Thank you. Yeah, do we have any questions from Ananda or Nick? You guys good?
I'm okay. I was just gonna request at some point just showing me things in the lab. I've said to you before, like I've done time in a lab, but I don't know like necessarily where all the stuff is and what it does. So I can't do that from here, but I will ask at some point.
Yeah, yeah, like, I don't know everything about the lab. I mean, there's, Gina and George know there's times where I'm like, where's this thing? And um yeah, I think, and as well as like, different, like I mentioned, like the chemicals and stuff. There's just some very, very specific things about very specific chemicals. So, I'm not that experienced. I'm not gonna know about everything. So, but it'll be like, we figure it out together, and
Mm.
as long as we adhere to those basic principles, it should be okay. Yeah.
I need to write down
Like,
the autoclave process as well. Like
Oh,
yeah,
true. Yeah, we should definitely have like a manual for each of the
or a
equipment
sticker on the front of it or something.
yeah, that'd be important for sure. Sure. Yeah.
yeah. The way I kind of see, or that I think that we're envisioning this safety lecture is just sort of a way to introduce people to the lab and make sure that everybody knows the basic procedures of how to be in the lab safely. And from there, we can all work together and help other members to learn how to use the equipment and how to use it safely. Yeah, and so we can coordinate that among ourselves, no problem. Mhm.
Would it be possible if I did the exam later on in the day and not right at this moment?
That's fine. Yeah,
Yeah,
that's
yeah.
totally fine. Whenever is convenient for you.
Okay, thank you.
Yeah, and then for Nick, who already has lab access, and people who are watching this in the future who may already have it, please still complete the quiz. This is a way for us not only to make sure, of course, people know how to use the lab, but because we want to be very exact and good with our record keeping from here on out, with knowing just who has access to the lab. That way, in case there is any incident in the future, or we want kind of a, how do I say, like a count of how many people use the lab or how many people have actively expressed a desire recently to go in and use the lab for things, then we can at least have an accurate count. So although like we'll get your email address to set you up, those personal contact information will not be distributed publicly, of course. But yes, please take the quiz if you want to get or keep lab access. Thank you. Any final questions, comments, or concerns before we finish up the session?